Welcome to the Bedside Biology seminar series. Today, I want to give a very basic breakdown of CAR T-cell therapy. So with that, what are CAR T-cells? Well, CAR T-cell stands for chimeric antigen receptor T-cells. Now, honestly, that isn't very intuitive. However, if you break it down piece by piece, it becomes a lot more clear. So the first part, chimeric, you can really think of it as the mythological creature known as a chimera. And typically we see that in media presented as a fusion of a snake, a lion, and a goat. In biology, when we use the word chimera, we're also typically referring to a fusion of different species. For example, if we're talking about antibodies, we may generate the antibody in an animal host like a rabbit or a mouse, but we may actually take a portion of that antibody and fuse it with a human antibody so that we have a humanized antibody that we can then use as a therapeutic. And we're actually going to revisit this idea shortly. The next part, antigen receptor. It's really any receptor that recognizes an antigen, for example, antibodies. And an antigen is any sort of foreign substance that will elicit some sort of an immune response. So, for example, pollen is an antigen. And when springtime comes, many of us get sniffles, watery eyes, and this is really just symptoms of our immune response to the pollen. Now let's circle back to the example we were using earlier to really get at what a chimeric antigen receptor is. So if we think about generating those antibodies or antigen receptors in another host, like a mouse, we can really optimize them. We can get them very specific to our target of interest, perhaps to a tumor marker, for example. Now, the problem is if we go and we stick that antibody directly into a human, the human body is going to recognize the whole entire antibody as foreign. Our immune system will therefore mount an attack on the antibody itself and not let it serve its function. It won't actually get to the target that we raised it to. So instead, what we can do is just snip off the functional bit of the mouse antibody, the portion that would recognize the tumor marker, for example, and then we can fuse it to the backbone of a human antibody. And this is going to give us a more humanized antibody so that now if we were to put it into a human, hopefully it will not elicit the immune response and now can actually serve its function. It can go and bind to the target of interest, perhaps the tumor marker we've been referring to. So now that brings us to the very last bit of this word breakdown, T cells. Now, T cells are kind of rock stars of our immune system. However, when they very first start out, they're immature and they're not really functional. At this point, they're known as naive T cells. Now, for them to be considered mature and functional, they must actually express antigen receptors, right? They have to be able to recognize some sort of antigen so that they can target threats to our health. Now, can you imagine if we could tweak these immune cells to specifically recognize a target of interest like the tumor marker we've been using in our other example? What if we could tweak them so that we know they're very efficient, we know that the patient we're introducing these to is specifically expressing the antigen we've tweaked these to? That would be really, really awesome, and it would probably be very efficient. And that's exactly the idea behind chimeric antigen receptor T cells, is to tweak them so that they're very effective, but still our own immune system. So now I want to spend some time really breaking down why CAR T cell has been so effective. And to really answer that, we need to understand why T cells are rock stars. So before we really dive into the nitty-gritty about T cells, I just want to give some basics. So T cells are also known as lymphocytes, and they're a type of white blood cell. They handle infections that we haven't been exposed to. And as we previously mentioned, they start off as naive T cells until they encounter an antigen-presenting cell, which is going to help train them to recognize a specific antigen. Once they encounter an antigen-presenting cell, they can mature into 4 different types of T cells based on some other signals given. We're going to dive into each type, but quickly the 4 types are cytotoxic T cells, helper T cells, regulatory T cells, and memory T cells. So let's start off with cytotoxic T cells. You can think of them as the action guys. They're the T cells that are responsible for actually attacking the target. And they actually follow the most straightforward pathway as well. So let me go ahead and get my laser pointer. Here we go. T cells start out in the thymus, and when they leave the thymus, they'll either be stamped with a CD8 or CD4. Now, I know you may have heard CD8 positive and CD4 positive, but perhaps you didn't really know what that was referring to. All CD stands for is cluster of differentiation, and it's really a cataloging system of surface proteins. So, for example, if we were to discover a bunch of different types of cells that all happen to be purple, and they all happen to have a big surface protein in common, and we noticed that other cells that weren't purple didn't have that particular protein, then we would get to refer to that protein as cluster of differentiation, or CD, and whatever number the scientific community is currently on. So let's just say X. Then our CDX protein will become known in the scientific community as an indicator of purple cells. So let's get back to T cells. Any T cell that's destined to become a cytotoxic T cell will be stamped with CD8. So now, even though it's left the thymus, with the goal of becoming a cytotoxic T cell, it is still in that immature phase or naive phase. And you can think of naive in this context as we would conversationally, innocent even. And that's because it hasn't been exposed to any antigens yet. So in order for a naive T cell to really get schooled, it heads to a lymph node. Now in the lymph node, it will meet the antigen-presenting cells, which are kind of the bad influences. They're the ones that actually introduce that innocent T cell to the antigens. And once the T cell recognizes the antigen and is fully mature, then it can go ahead and enter circulation to find its target and be functional, and in this case, attack. So the next group of T cells that I want to briefly touch on are helper T cells. And they start out the same way as cytotoxic T cells. However, when they leave the thymus, they have been stamped with CD4 instead of CD8. Now, once they get to the lymph node to get schooled, they do have more routes that they can follow based on what is actually ailing the body. So, to help discern what kind of helper T cell the body needs, there will be cytokine cues, and these are all based on the pathogen that's circulating through the body. And that will give an instruction on what kind of T cell the naive T cell needs to mature into. For instance, if there are intracellular pathogens like viruses, that will cause IL-12 to be produced by the body, and this will tell the helper T cell to become a type 1 helper T cell, or Th1 cell. The Th1 cells will then release interferon gamma. And And what that does is it will actually recruit other immune cells to come and address that pathogen. And that includes immune cells like the cytotoxic T cell, as well as macrophages. Now, there are other classes of helper T cells, and based on the cytokines that it's exposed to, it will say which pathway to follow. For instance, if we have allergens or parasites in our body, we will be exposed to IL-4. This will stimulate the helper T cells to become type 2 or Th2 cells, and they'll also recruit other immune cells by releasing their own cytokines. So helper 2 T cells will release a lot of IL-10, and this will recruit granulocytes in particular. Now, moving along, the next class of T cells are regulatory T cells. And like the helper T cells, they're still stamped with CD4 rather than CD8. They will also be exposed to cytokine to cue them down the regulatory path. However, there's not any variations. And the cytokine that they're exposed to is TGF-beta. Once they receive the cue to follow the regulatory pathway, they themselves will start to release TGF-beta to recruit more regulatory cells, as well as IL-10. And this will ultimately calm down the T cell response. So they act as the feedback inhibitor, because we really don't want our immune response to get out of hand or to continue long after the threat has been eliminated. And the final group of T cells that I touch on are memory T cells. Now, much less is actually known about how memory T cells are regulated and maintained, but they act as our circulating pool of primed T cells so that they can respond to known threats immediately. And because they're a primed pool, it includes both CD4 and CD8 positive T cells. So, getting back to the heart of the question, why is CAR T cell therapy so effective? It's because the T cells are rock stars. They really have all bases covered. We have a group of cells that can really take action are cytotoxic T cells. We then have another subset that are there to help the cytotoxic T cells recruit other immune cells, and those are helper T cells. Then we have a system in place to keep this all under control. We have the regulatory T cells. And in addition to all three of these, we have the memory T cells, which give us a group of cells that are ready to go if there's a known threat that's recognized. So if we bring it all back to the idea of it being used as a therapeutic, I want to go over just a few more basic details as applied to a therapy. So CAR T cells can be both CD4 positive and CD8 positive. And ideally, we really want a 1-to-1 ratio so that we're getting the full package. The next point, it was not intuitive to me and was very intriguing to me, and that's that T cells can be harvested from patients or healthy donors. Now, traditionally, when we talk about T cell therapy, we think of it as coming from the patient, and that's the ideal circumstance. But now, as we're starting to think of applying this on a global scale, we need to face some of the roadblocks that would come with this therapy, and that's that we would be using this in cancer patients, and many of the cancer patients have depleted immune cells. They may not have the best population to harvest from. And as we mentioned, we are genetically modifying these cells so that they can recognize a very specific feature, oftentimes a tumor marker that isn't expressed in normal tissue. And because T cell therapy is ultimately using our own immune system, the most common side effects are from our immune system ramping up a little bit too much. For instance, cytokine release syndrome, which can be very severe. However, under close monitoring and rapid response, this can also be managed. Now, I'm very happy to touch on the final point here, and that's that the initial response rate is usually very high, in the 90s range, that patients respond to the therapy. Now, unfortunately, the long-term remission and survival rates are lower, and that's because of antigen escape. And antigen escape is a very important concept, not just to CAR T-cell therapy, but also to understanding how tricky treating cancer really is, which is why I dedicate another seminar to focusing on both antigen escape and tumor heterogeneity. So please tune in for that seminar at your convenience. So with that, I want to thank you all for taking the time to tune in to my seminar, and I hope to see you back again. Thank you.